The overall goal of this procedure is to evaluate the effects of tobacco product preparation on cytokine secretion and target cell killing by PBMCs. This is accomplished by first treating cultured PBMCs with whole smoke conditioned medium or nicotine, and then intracellular staining of the treated PBMCs is performed to determine their cytokine production. Alternatively, CFSE-labeled K562 cells are co-cultured with whole smoke-conditioned medium-treated PBMC effector cells, and the viability of the K562 cells is qualified by 7-AAD staining. Ultimately, the effects of the combustible tobacco product preparations on the PBMCs from either assay are measured by flow cytometry.
The main advantages of our techniques over the existing methods such as chromium release assay or ELISpot are our methods are high throughput, non-radioactive, and we can measure the cell functionality.
Though this method can provide insight into the biological effects of tobacco product preparations, it can also be applied to the evaluation of other compounds that impact the toll-like receptor-mediated and cytolytic functions of PBMC effector cells.
Before beginning the staining procedure, dilute whole smoke conditioned medium and nicotine at the listed concentrations in RPMI complete medium to a total volume of 100 microliters per well in a 96 well plate.